understand the main aspects of the anti-cancer immunoglobulin. I remember we couldn't do immunoglobulins. Yeah? So we will do them after we have done the immunosuppressant. Okay? It will become easier for us to relate to them. Okay? Please call your colleagues, whoever are around, or do you have a lecture after this?
Yes, physiology. Physiology? So it's a review lecture.
It's 11 to 12.
Yeah. It's not.
Okay, guys. Amar, have a seat, please.
Yes.
There's no more cross-linking or whatever you call it. Cross-linking.
GG.
Okay, so let's start.
Last lecture.
The remaining slides, so the last one we would do after we are done with the current ones, because it will make us at ease. Okay? There will be a lot of cross-reference, so it will be easier. Okay, so let's start with immunosuppressants and modulators. Now, today we are discussing, two different classes of drugs. Immunosuppressants, as the name indicates, suppress, hamper, retard the immune reaction, immune response, okay, to any antigen, or even without an antigen. Whereas the immunomodulators do not necessarily be the opposite, okay, but they are the drugs which take part in bringing about some aspects of the immune reaction, okay. Sometimes they fine-tune the immune reaction, sometimes they support the immune reaction. So they do a number of jobs which cannot be accommodated either as immune boosters or immune suppressants. So it's actually a multitude of factors, that's why they are called as the modulators rather than boosters or suppressants clearly. Okay? So immunosuppressants are the drugs which clearly suppress the immune function, and the modulators, immunomodulators are the ones which sometimes they are used to bring about the immune reaction, sometimes they are used to support, and sometimes do some other related jobs. We will see. Immunity, is either innate or adaptive or acquired. The innate is the one which is inbuilt by default, okay, non-specific, generic, okay, in anyone's own body, whereas the acquired is the one which is earned, which is made, okay, it's newly synthesized, made in due course of time. So from the very beginning it doesn't exist, but as the time passes by, it evolves, okay. So acquired immunity could either be pertaining to the active immune response, which depends on the active production of antibodies, okay? Or it depends on the antibodies, the ones which are transferred from outside rather than synthesized in body, okay? So it's passive. So either it's active, that means producing actively their own antibodies, or it is passive. That means it doesn't refer to the own antibodies, but but antibodies obtained through some other source. Okay. So again, now this is a dichotomous classification. So active own antibodies could either result from natural exposure to any antigen, or here it's infectious agent, written infectious agent, but it could also be non-infectious antigen. So it could be any antigen obtained from the natural environment, whereas the artificial is the one which is induced, okay, through immunization or vaccination. The passive, similarly, could also be natural, transferred naturally. For example, during the pregnancy, the fetus receives some immunoglobulins from mother. Okay? And after delivery, the newborn, if breastfed, receives some antibodies in milk. Okay? So those are natural antibodies, but passive. And the other ones are artificial, though passive but artificial. Okay? For example, if you remember the road traffic accident example, during the emergency, if the road traffic accident takes place or any contaminated injury takes place, the tetanus protection is given in the form of preformed antibodies injected. Okay? So they are injected. Similarly, anti-venom. Okay? If there is a snake bite, you give anti-venom. Okay? So there are preformed proteins to given for the immediate production rather than the waiting for synthesizing the immune response. So it's a dichotomous classification of various immune responses. Now, and you know that in all the active immunization or active immunity, the CD4 positive cells, the lymphocytes, the CD4 positive, they play the regulatory role. They have a central role to play. I hope you remember. Where they act, this is the green cell here at the border, is the between the white and the pink areas. This is the CD4 positive cell, which receives the processed antigens from the antigen presenting cells. Okay, for example, dendritic cells, macrophages, Kupffer cells. Okay, so those oligodendrocytes, all of them are
antigen presenting cells.
Antigen presenting cells. And those antigen presenting cells, they engulf the antigen, modify it little, so this is called processing of the antigen, okay? And then, along with MHC, they present it to the T lymphocyte, CD4 positive T lymphocyte, and which is then responsible to mount the immune response, as well as create memory cells, okay? So the memory cells are either created here, or it induces the T cell or a B cell response. Clear? Is it clear?
Yes. Yes. Sure.
Okay. And here you can see some important mediators like TNF alpha, interferon gamma, interleukin 4, and you know for interleukin 2, which stimulates and recruits other T helper cells. Okay, so T cell communicate with other cells through certain chemical mediators. We call them cytokines. Okay, will I move on? Okay. Further detail of what T cell is doing. In front of you is a T cell, which is bestowed with many recognizing receptors. You can understand this is a regulator, so it has to have a number of receptors for different interactions, different types of interactions. In front of you is a schematic diagram of an antigen-independent activation. This is literally chemical activation. Okay? See, cytokines or free radicals, they are able to stimulate T cells so that they can recruit the immune cells and bring about the immune response. So this is antigen-independent activation. Okay? So far we knew this is antigen-dependent, but now we are adding to it. This is antigen-independent. This is a chemical stimulation of the T cell. Okay? That means what we studied before, that could be classified or grouped as antigen-dependent activation, which depends on processed antigen presented by some antigen-presenting cell. Okay? And antigen-presenting cell has got a number of molecules. It's a sort of a pattern, okay, which is then recognized by the T cell, okay, with the corresponding molecules. And this pattern, once recognized, remember those video games which ask you to provide a pattern of the key. Once the key and the lock, they match, the downstream or the further stage is attained, in video games. Similarly, it's just like the analogy. Once the pattern is recognized, it is stimulated and the immune reaction sets in. If the pattern doesn't, get recognized, the immune reaction is not mounted. Okay, this is one of the ways how the foreign antigen is subjected to the immune reaction, whereas the self-antigen, they skip that immune reaction most of the times. Okay, so let's see what immunosuppressants we have. And now, given the central role of T lymphocyte, we can assume that many of these drugs would target the lymphocyte, of course, the T lymphocyte. So let's see. Glucocorticoids, you know it from the past knowledge and the very immediate PBL, okay, done this week. Then you have mycophenolate mofetil, which is also abbreviated as MMF, mycophenolate mofetil. Then you have a whole group of related drugs which are immunophilin dependent. We will see what immunophilin is and what these drugs have to do with this. For you, there are only 3 drugs in the list. The blue ones are the ones which are in your drug list, which is present in the guidebook, the course book available on Moodle. Okay. Yeah. And cyclosporin, tacrolimus, which is also previously been called as FK506, sirolimus, which was previously called as rapamycin. And then you have monoclonal antibodies. We have few monoclonal antibodies as part of your drug list. Okay, then you have cytotoxic agents. If you can pay attention, azathioprine is the only new agent here. Methotrexate and cyclophosphamide were the ones which you did as an anti-cancer drugs. There are some anti-cancer drugs which in low doses work as immunosuppressants. Okay? And 2 examples, rather 3 examples are shown here. Cyclophosphamide and methotrexate, okay, and azathioprine. Okay. So, let's now see what immunomodulators you have. Immunoglobulins, okay? So, for you, you have only a few examples out of the long list of immunoglobulins. Alemtuzumab, basilixumab, daclizumab, muromonab, and antithymocyte globulin. Okay, these are the ones which you should know. And here are certain viruses and disorders where they are utilized. Okay, then you have other immunomodulators like interferons, gramazole, thalidomide, and BCG. Remember the BCG is a vaccination which we get for tuberculosis. Okay, but there is more to BCG than